We will go ahead and get started again. So as you see here, it's a little bit different from what I said this morning. It wasn't by, I didn't do it on purpose, but there were some difficulties to doing exactly what we wanted. So to move one whole session in the front and then the other session in the back. So we're going to do a little bit of both. So we're going to start with the session 1.3a, which is connecting microbes to metabolism using gnotobiotic models. We will hear presentations by Federico Rey and Patrice Cani. And they will have, after they finish, there will be a specific Q&A time allotted to this panel. When they are finished, around 2 o'clock, we will move to have the whole of session 1.2, as it is in your book. First, Robert Britton, followed by Vincent Young, and followed by Donald Ingber on the web. We will have a break, and we will finish with the 2 sessions of session 1.3b, 1.3c, which remain in their allotted time slot. I apologize for this, but as I said this morning, we needed to accommodate flights, and so this was the best we could do under the circumstances. So thank you very much. Dr. Guillemin has very graciously agreed to moderate the session 1.3 A, B, and C, and Dr. Young will be speaking and moderating session 1.2. So please, thank you.
Okay, so we'll be having this first session, connecting microbes to metabolism using gnotobiotic models, and Federico Rey will be our first speaker. Thank you. So this is your that's the pointer.
Okay.
And
How do okay, that will
yeah, and then I think the best is to use this. The timer should go.
Is okay? Everybody hear me? Yes? Okay. Good afternoon. My name is Federico Rey, and I'm going to talk about connecting our work in trying to connect microbes to metabolism using gnotobiotic mouse models. So we are exposed to thousands of microbially derived metabolites, and these metabolites are generated as product of metabolism of microbes that inhabit our body. So when microbes make a living, they generate building blocks, they respire, they generate energy. They they generate metabolites that impact our physiology. Microbes also have what is called secondary metabolism. And very generally, these are metabolites that are not necessarily linked to growth, but that have effects on fitness and the ecology of the ecosystem. And within this category, we can put biotransformations of many dietary compounds that microbes do, or drugs, or even host metabolites like bile acids. So we know that bile acids, for example, are heavily metabolized by microbes in the gut, but we don't really understand why microbes do this. So what we know is that many microbial metabolites affect community dynamics or have dedicated host receptors that allow for microbe host crosstalk. And so I'm interested in the interactions between gut microbes and diet. And so the diet that we consume provides a lot of substrates for microbial metabolism. So it's good that we just have lunch. So this is, but when you eat if you eat your salad, you're feeding your gut microbes a set of complex polysaccharides that we cannot process with our host, with our human enzymes, and reach the distal gut, where are metabolized or fermented by by microbes and converted into short-chain fatty acids, such as acetate, propionate, and butyrate. These metabolites affect the motility of our gut. They modulate they affect the histones and regulate gene expression, and they have broad effects on us, like they protect against diet-induced obesity, or they even modulate blood pressure. I am from Argentina, and Good Argentina has lunch with a good Malbec. And red wine is a great source of polyphenolic compounds or flavonoids. So flavonoids have been linked to everything. They're supposed to be beneficial for everything or for nothing, depending what paper you look at. But something that has been ignored in the field of polyphenols or flavonoids for a long time is that flavonoids are not absorbed as they're present in the food that we consume. They're rather heavily metabolized in the gut. And they're the products of this microbial metabolism what affects our physiology. Now, if you sort of complement your meal with a good steak, you're consuming not only a lot of protein, but a lot of sort of choline and carnitine. And these are essential nutrients for us. But there's also gut bacteria that ferment these substrates and generate compounds that have been associated with disease. So from what we heard this morning, we know that there are large interpersonal differences in microbiota composition. So what that means is that if you were to eat this meal that I have here in the background, you will all generate different amounts and different proportions of all these different metabolites. And so I'm interested in how our microbial differences modulate the nutrients that we get from the food we consume and how this affects our health. So understanding this would allow us to maybe personalize nutrition based on the metabolic potential of a person's microbiota. But before we can get there, we need to understand who's doing what. We have hundreds of microbes in our gut, and so if we're going to try to do any personalized nutrition, we need to understand what each one of these 100 species are doing in our gut. So in trying to do that, we are faced with the challenge of the high complexity and the lack of resolution that our microbial communities have. As I say, hundreds of species in our gut. So the approach that my lab takes is to colonize germ-free mice with species that are representative of the native community and its phylogeny and function. So we basically have a defined microbial community that consists of all sequence isolates of the human distal gut. And so this eliminates the uncertainty surrounding who's there. We know exactly who's there because we grew these organisms and we use them to colonize the mice. And with this model, we can easily manipulate the composition of the microbiota to test hypotheses. And we can even do in some cases, like the one I will describe today, we can do microbial genetics in some of these species in the gut. So I will tell you about our approach to study micronutrient interactions in the context of the work that we've done with choline, an essential nutrient. And this has been a collaboration with the laboratory of Emily Balskus and Daniel Amador Nogues at University of Wisconsin, and a student in my lab, Kim Romano, that's done most of this work. So choline is an essential nutrient that is very important for us. But it turns out there are bacteria, and this has been recognized for a long time, that there are bacteria in the gut that will take some of this choline and ferment it to trimethylamine. And then that trimethylamine is absorbed by the host and converted in the liver into trimethylamine oxide. And the enzyme that does that is called flavin monoxygenase, and that is in the liver. So in 2011, this metabolite gained a lot of attention and was put in the spotlight because of some of the work done in Dr. Hazen and Dr. Lucius' lab suggesting that the TMAO is a good predictor of cardiovascular disease in humans. So the higher the levels of TMAO in your plasma, and this is, in thousands of humans, they look at this, the higher levels of TMAO, the higher the chances that something really bad is going to happen with you, basically. And so this is just, and they follow this up with subsequent experiments in mice, where they fed the mice TMAO, and they showed that TMAO exacerbates, so it wasn't just associated with disease, but it was causally linked with the development of atherosclerosis in mice, and sort of follow-up, mechanistic studies show that TMAO inhibits reverse cholesterol transport and contributes to platelet activation, and in that way exacerbates the development of atherosclerosis. So since that study, there have been many more, and so TMAO is kind of a hot molecule these days. And so many studies showing association between levels of plasma TMAO with a number of different diseases, including diabetes, renal failure, heart failure. And except for heart failure and atherosclerosis, I think that in most cases, these are just associations. The causality is not clear. So I started my lab 3 years ago, and some of the questions that we have is, we're interested in learning about what are the microbes in the gut, or the human gut-associated bacteria that use choline, and then which ones of these species that use choline converted into TMA. We were also interested in understanding how microbial choline metabolism and TMA and TMAO production affected host biology. So the first approach we took was to screen a collection of human gut isolate, a collection of about 100 different organisms that we can grow in the lab, so it's representative of the diversity seen in the human gut. We can grow them anaerobically in 96-well plates with labeled choline, and then we use mass spectrometry to measure the levels of choline left in the media and the amount of TMA produced, and in that way identify the bacteria that metabolize or choline into TMA. By doing that, from the hundreds of species that we screened, 8 came out positive. And something that I want to emphasize, that only these 8 species consume any of the choline that we put in the media. It seems that more than 90% of the bacteria that are in our gut do not metabolize, or they're not they don't use choline for metabolism. It turned out that 7 of these isolates that, so that most of these isolates that we identify as TMA producers encode for a pathway that was discovered by Emily Balskos. So in this pathway, choline is converted by a choline TMA lyase into TMA and acetaldehyde, which is then detoxified and converted to acetate. But sort of, it seems like this is the main pathway in the human gut, the most common pathway. There are other pathways that occur because some of the organisms that we identify as TMA producers do not encode any of these genes, and we are sort of interested in sort of identifying these new pathways. But we were quite lucky because by doing some in silico work, we found that there was an E. coli strain associated from the Human Microbiome Project that had this pathway. And so, if you think as a bacterial geneticist or microbial physiologist, having the ability to do genetics allows you enables you to investigate the function of different genes. So we can create mutants in the different in the gene that is essential for the production of TMA, the choline TMA lyase, and then we can generate a strain of E. coli that is unable to generate TMA from choline. So by having this strain is very useful for us because we can use gnotobiotic models to ask how does the choline metabolism or TMA production affect host biology. So we turn into our gnotobiotic mouse model, and we created two different communities. So we created a community that consisted of 5 different organisms, none of which are able to metabolize choline. In addition to that, we add the E. coli wild type strain that I just told you can convert choline into TMA. So that is the choline consuming community. So having that wild type E. coli allows this community to convert choline into TMA. The other community contains the same 5 species plus the CAT-C mutant that cannot generate TMA from choline. So we have 2 communities that vary only in one gene, and that is the gene that conveys the capacity to convert choline into TMA. So when we measure the plasma levels of TMAO, so what circulates in plasma, in the mice, we see that only the mice that are colonized with the wild type E. coli plus the core community accumulate TMAO, as you would expect. But something that to me was extremely interesting was that the mice that were colonized with the choline consuming community had much lower levels of choline than the mice that were not able, that were colonized with the community not able to consume choline. And so this is, I think, is important given that choline is an essential nutrient, and we are not considering choline. We're not considering the gut microbiome when making recommendations about the needs of choline consumption. So choline is important for many functions. Among them, it serves as an important methyl donor. So you go from choline, and these are host functions. Choline is transformed into betaine, methionine, and to S-adenosine methionine, which is the universal donor for DNA methylation. So we look at all these metabolites in the plasma and in the liver or mice colonized with these 2 communities. And what we found was that being colonized with a community, with the microbial community that consumed choline, lower the plasma levels of not only choline, but all metabolites involving 1-carbon metabolism. We thought this was relevant and interesting, so we decided to pursue this. And we look at the DNA methylation levels at a number of different tissues, including liver, colon, brain, and heart. And we found that, again, that having the mice that were colonized with the choline-consuming community have much lower levels of DNA methylation, suggesting that the microbes are competing with the host for choline, and and in that way they lower the viability of choline for the host and the viability of methyl donors, and resulting in difference in DNA methylation. Okay, so high-fat diet exacerbates the need for methyl donors and choline in different tissues. So we decided to test how these 2 different communities that I mentioned before will affect metabolic disease in mice fed a high-fat diet. So, we saw that the mice that were colonized with 2 different communities, 8 weeks on high-fat diet, they have the same weight. But then mice that have a choline-consuming community have much fat, accumulated more fat, and they accumulated much higher levels of triglycerides in the liver compared to mice that were colonized with a community that cannot metabolize choline. Choline is also very important during pregnancy, and a number of studies have shown that choline deficiency during pregnancy has detrimental effects on the offspring. A recent study show that 40 months sort of after the kids were born, that children that had received choline supplements in utero and after birth had fewer attention problems and less social withdrawal than those mothers that consume an extra amount of choline during pregnancy. So here we're comparing two groups of mothers that have consumed either the recommended amount of choline or an extra amount of choline. And so more choline seems to be beneficial in this, for these kids. So we decided to test whether colonization of the mothers affected the colonization status and choline consuming status of the mothers affected the offspring DNA methylation level. So here I'm showing you the plasma levels from the mothers, the plasma level of TMAO, of mother colonized with a TMA producing or choline consuming TMA producing community or not. And so only the mothers that have the choline consuming community accumulate TMAO. And as I told you before, mothers colonized with this community have a lowest level of choline. What we did was when the mothers were ready to deliver, we took the pups by C-section and we look at the levels of methylation in the brain of these pups. And we see that the DNA colonization status of the mother basically affect the levels of DNA methylation in the brains of the pups at the day of birth. Now, so in this case, the pups were sort of sacrificed or euthanized at the day they were born. But if we were to keep these pups, there are two things that happen. One, the mothers eat half of them. They tend to cannibalize more frequently the pups that mothers that are colonized with a choline, with a community that is not able to consume choline. And we also perform an assay that is sort of supposed to tell us about a behavioral assay called the marble bearing assay that is supposed to tell us about the anxiety and obsessive compulsive behavior that these animals have. And again, we see that mice colonize. So basically in this study, you present the animals with marbles, and the animals tend to bury some of the marbles. But the more stressed or the more anxious they are, the more marbles they bury. So again, here the pups that were colonized with the choline-consuming community, that were born from these mothers, buried pretty much all the marbles, whereas there was a wide variation in those pups and lower levels, of bearing for those pups that or animals that were colonized with the community not able to consume choline. So in summary, I told you that I didn't go into much detail about this, but the microbial choline utilization pathway is widely and discontinued distributed among gut bacteria. So the 8 species that we identify as choline-consuming bugs are represented across different genera, and we even find that two different strains of the same species, one can metabolize choline and the one can't. So we cannot predict this sort of metabolic activity by 16S. I told you that microbial choline-dependent TMA-producing pathway results in lower availability of choline, downstream metabolites, which are key for methyl donation. We see lower levels of DNA methylation in multiple tissues, increased adiposity and triglycerides, and possibly alter behavior. So we are exploring more the behavior findings that we got. So I feel that those are quite preliminary yet, still. So one conclusion is that the microbial choline utilization pathway may limit choline availability during pregnancy and affect brain development. And I think this is something to start thinking about, because current dietary recommendations do not consider the interpersonal differences in choline consuming bacteria in the gut. This will be most of my talk, but then I have 2 slides that I was asked to comment on the challenges in the field. And so these are sort of some of the challenges that I find that sort of we have in the field. So we have, thousands of microbial metabolites, but it's hard to decide which ones are biologically relevant and how to prioritize their study. So in the case of the, the Choline story, for us it was sort of inspiring the work that Dr. Hazen and Lucius done in that field, suggesting that TMAO was an important metabolite to study. And so that's how we choose to, how we prioritize. But I think that's in general. We know that there are a lot of chemical diversity coming from our microbes, but which metabolites are the relevant ones. And then a big sort of, I think, I show you how we try to link specific metabolic microbial activities with host biology. That was one example. I'm not claiming that it's the only way to do it or the best way to do it. That's how we are trying to link it. But something that it was very helpful for us to move forward with this study is the availability of one strain of E. coli that happened to have the pathway that we're interested in studying. We were kind of stuck before we found that strain because the truth is that there's a complete lack of genetic tools to modify commensals. And so, and that I think is very important to move sort of, for understanding microbial or bacterial metabolism at least. In terms of the welfare issues, I think that sort of it'd be nice or be helpful to have access to gnotobiotic resources, especially genetic mouse models, that different facilities or from different universities can have access to reduce the number of animals produced but not used by individual investigators. So in the future, we're planning to do mouse work with germ-free, with strains of germ-free mice or different knockout species, and maintaining all those strains germ-free. One is a lot of work, and two will result in sort of having to keep mice for long periods of time and have to use euthanasia without being used. With that being said, I'll thank people in my lab and funding. Thank you so much for the invitation.